Hi, good morning, everyone. Welcome to Microbial Minutes for March 26, 2018. This is the American Society's every couple week update for what's been going on in the microbial sciences. I'm Julie Wolf, and here today to talk about a number of things that have been going on in microbiology and science. So we have a couple different research articles and some policy updates. We're going to talk about the effect of some non-antibiotic drugs on the gut microbiome and the use of artificial intelligence to discover new viruses. We will talk about hybridizing the lipid properties of archaea and bacteria and what that might mean for the origins of life. And then we're going to talk about a fun article that discusses the potential for spreading disease on an airplane, which seats are most exposed to patients or travelers that might make you sick. Finally, we'll end with some updates on the new congressional budget that was passed just this last Friday on March 23rd. So let's dive right in. I'm going to be answering questions, so go ahead and put those into the chat. I'll check them at the end. I think that might work a little more seamlessly, so feel free to participate. And if you're listening to this after the live session, feel free to leave a comment and we'll check back and answer whatever your questions may be. All right, diving into the first report. This was from Nature. It was titled Extensive Impact of Non-Antibiotic Drugs on Human Gut Bacteria. The take-home message is that a quarter of non-antibiotic drugs affect the gut microbiome members, which is a huge number when you think about these drugs that are not antibiotics. So these are drugs such as chemotherapies, but also things like painkillers or psychiatric drugs that have been tested for their ability to target either human molecules or human processes within our cells. The authors wanted to look at the question of how many non-antibiotic drugs affect the growth of at least one bacterial strain of the microbiome. And so they looked at lots of classes of drugs to study their effects on microbiome members, but of course, things that are antimicrobial like antiprotozoan, antifungal, antiparasitics, and antivirals is shown here, and antibiotics up here. Those are classes of drugs that one might expect to have an effect on different members of the microbiome. What this group was concentrating on were these human-targeted drugs. So those are things that are supposed to target specific organs or specific types of human processes, as I mentioned, things like our ability to detect pain. And so they looked at 835 drugs and found that over 200 of them can potentially affect microbiome composition. And so what they did was to measure the effect of each of these drugs in a large-scale screen against members grown in vitro, singularly, in the presence of this drug. And they looked for the inhibition of growth that might be conferred by the presence of different drug concentrations. Now, in this type of large-scale screen, they're not only screening over 800 drugs, they're also screening several dozen different bacterial strains and species. It's hard to look at different concentrations of drugs. The researchers in these types of programs generally choose one concentration of drug, and that can be problematic, as the researchers themselves address. So in this figure from the supplementary data, they show what happens when they looked at metformin. Now, here they have along the x-axis all these different bacterial strains. They may look familiar to you, things that are part of the gut microbiome, such as E. coli or Bifidobacterium or Lactobacillus, Fusarium, all these different species, several of which we've talked about on different sessions of Microbial Minutes. When they looked at their screen, they used a 20 micromolar concentration of this metformin drug. It's a drug that treats patients who have diabetes. And they looked at the inhibitory concentration to inhibit 25% growth. And at 20 micromolar, they saw no inhibition of any of the strains or species that they tested. However, metformin within the body is not equally distributed at 20 micromolar and can increase to up to 1.5 millimolar in the small intestine and the colon, which is, of course, the location of most of our gut microbiome. And when they tested this concentration, the 1.5 millimolar, they saw that there were 3 different species that were affected, that had decreased growth below that 25% threshold. And this demonstrates that there might be a concentration-specific effect that is undetected in their large-scale screen. This demonstration with metformin in particular shows the importance for looking at causation and not necessarily correlation, as many microbiome studies do. So we've talked a lot about different compositions of microbiomes that might be, for example, in this example, the microbiome composition of diabetic patients versus healthy individuals. And if there is a difference that's seen, it may be assumed that that is due to the different processing of the diabetic patients, of their blood glucose levels. However, what this suggests is that it's important to take into consideration treatments such as metformin therapy that may affect that composition. And this type of causation study, where they actually add the drug in and show that that has an inhibitory effect on growth, help to determine whether it would be the drug itself or perhaps the ability of the patient's, the availability of glucose within the patient that could contribute to those different compositions. Now, this is just one example, and I mentioned that over 200 actually had an effect on the gut microbiome on some of these different species, which was a big surprise to many researchers. So this was shared quite widely on social media by different researchers, and it was written up in The Scientist, where a quote from one of the co-authors stated that the shift in composition of our gut bacteria contributes to drug side effects, but might also be part of the drug's beneficial action. And this is actually a positive spin from the study, where the authors suggest that some drugs that are not thought to be antibiotics may be able to be used for their antimicrobial effects. In particular, drugs that inhibited the growth of C. difficile, which can be a really potent infection in certain patient populations, may help to control the growth of that bacterium in patients who need treatment and are failing to respond to antibiotic therapy. So it gives a potential for new antibiotics. However, the flip side of that is that some of these non-antibiotic drugs may actually select for antibiotic resistance. And this was written up in CIDRAP. This is the Center for Infectious Disease Research and Prevention, an institute at the University of Minnesota. Here, this is an institution that focuses a lot on antibiotic resistance, and they stressed the finding that some of the E. coli that overexpressed genes related to antibiotic resistance were also overexpressed in the presence of some of these non-antibiotics. So this is not something that has been definitively shown, but it lends to the suggestion that some of these non-antibiotic drug uses may actually select for resistance within the human microbiome. So I'm sure that there's a lot more to come out of this study. Moving on, we want to summarize a research symposium that was held. The Joint Genome Institute had a few different presentations that talked about the use of artificial intelligence in order to find new viruses. The take-home message from these presentations is that artificial intelligence has found over 6,000 new viruses. And this is a problem or a new way to identify viruses that is going to be very useful, because viruses, unlike bacteria and fungi and protozoans, they don't have a similar DNA or RNA sequence among all viral genomes. In bacteria, we can look at the 16S ribosomal RNA gene in order to amplify and then sequence what might be there. In fungi, we can look at the ITS sequence and identify whether or not there is a fungal species that's been previously identified or not. But viruses don't have the same required gene that is shared between all different viral species. And so what some researchers have been doing is by teaching machines, giving them algorithms that have certain parameters that are common to different viruses, and then using those parameters to search through large sequence databases in order to see if there are new sequences that might fit into those parameters. So there were two presentations, one from Simon Rue and one from David Amgarten. And in one of them, Simon Rue's group was looking at Inoviridae. This is a viral family of bacteriophage. Those are the types of viruses that infect bacteria. And the computers were trained to identify genetic sequences that were unique to this particular group of viruses. And then the algorithm was fed a number of different metagenomic data sets. So these were sequences that had already been generated from mixed microbial communities. And using those parameters, the computer was able to find more than 10,000 genomes. In fact, potentially more than the symbol family that they thought that the Inoviridae were. And in the other presentation, there was a machine learning that was applied in order to find particular viral characteristics such as gene density. So viruses tend to have very dense genomes with little non-coding regions. And so using that characteristic and others, the researchers were looking at compost piles to identify new viruses from the local city zoo. Oh goodness, I forget which city it was. But both of these presentations are potentially going to lead to the newest iteration of a viral genome identifying software. And so this is both studies were kind of working off of the idea of a previous paper that was published in 2017 in BMC Microbiome, which was titled VirFinder, a novel K-MER-based tool for identifying viral sequences from assembled metagenomic data. And VirFinder had been the previous cutting-edge algorithm, which was used to identify viral sequences using some of these characteristics, such as gene density and nucleic acid sequences that were somewhat conserved among different families of viruses. And VirFinder itself had actually been an iteration of the VirSorter, a previous gene finding or viral genome identifying classification tool, which VirFinder was quite a bit more proficient at both identifying and at categorizing these different viral sequences. And so what the hope is that some of these presentations from the Joint Genome Institute meeting will lead to the next iteration that is yet more proficient at finding these viral sequences and give researchers a way to identify viruses that are hidden among the different metagenomic data sets that people have collected or are collecting. And so this was written up in a Nature article. Of course, all of our links are below the presentation. You can read it for yourself. One of the authors of the VirFinder article stated that previously people had no method to study viruses well. Now we have tools to find them. And those tools are going to be able to be used in clinical senses. For example, in patients who share syndromes that have no known etiology, we might be able to look through some of their microbiome or genetic data in order to identify viral sequences that were previously not identified. And so I do expect a number of papers to come out of these presentations. So stay tuned to future Microbial Minutes sessions, and we can hopefully talk about some of those new viruses that have been found. Moving on, the next paper to discuss is from the Proceedings for the National Academy of Sciences, or PNAS, titled Converting Escherichia coli into an Archaebacterium with a Hybrid Heterochiral Membrane. So the take-home message from this paper is that the E. coli lipid experiment gives insights to the LUCA membrane. So LUCA, the last universal common ancestor, is the progenitor cell to both bacteria and archaea. And here I want to highlight up here in the upper right-hand corner, we have a tree of life. This is a slightly old version of this tree of life, but it gives a sense of what where this LUCA was. So up here on the right-hand side, you can see that the archaea branched off, and over here on the left-hand side, we can see the bacteria branched off, and from the archaea, there were some Potentially, this could be the eukarya that are branching off from the archaea. But back before all of these different types of cells had differentiated, there was a progenitor cell, presumably, that was the LUCA, or last universal common ancestor. And the question is what is the membrane of this LUCA cell? And that question is posed because the archaeal and bacterial cell membranes have different shapes. They have different types of phospholipids that they incorporate into their lipid membranes. So if we look down here on the left-hand side, we can see the archaea have a glycerol 1-phosphate. So if we look at the glycerol head group of the archaeal cells, we see that the carbons, which are numbered as organic chemists like to do with their carbon compounds, we see 1, 2, 3, and it's on this first carbon where there's a phosphate group that's attached. But if we look at bacteria, we see it's on the 1, 2, 3, the third carbon where that phosphate's attached. And so that means that enzymes that either synthesize or recognize these different lipids would have to recognize a different shape of these hydrophilic head group here. And then the fatty acid chains are actually linked in different ways as well, with an ether linkage in the archaea and the ester in the bacterial cells. So what was the membrane of the last universal common ancestor? Was it more like the archaeal or more like the bacterial? Or was it a mix? Was there even a membrane at all? You may recall a previous microbial minute session, we discussed the idea that at some of those thermal vents that are down in the deep ocean, perhaps some metabolic reactions eventually led to the formation of lipids around the common reactions that could eventually become something resembling glycolysis. And so whether there was a membrane at all is a question. However, there are some conserved membrane proteins, such as ATP synthase and others, that are found in both bacteria and archaea, suggesting that this was something that was found in that LUCA cell. And so it's possible that there was a phospholipid bilayer in LUCA that then differentiated when the archaea and the bacteria separated. And so another possibility is that the first lipid synthesis enzymes had very poor specificity, and they couldn't really differentiate between that glycerol 1-phosphate or glycerol 3-phosphate backbone. That is relatively difficult to test. We don't have 3 billion years, which is how long ago that original cell was thought to exist. But scientists can look at what happens when archaeal lipids, this G1 phosphate backbone, is expressed in a bacterial cell, such as E. coli. This has been something that researchers have been attempting to do for a number of years with not very much success. And this group was actually able to increase the number of archaeal lipids quite a bit and look at their effects inside of the E. coli, answering questions about whether or not that was a stable membrane and whether or not that E. coli cell would even be able to survive and generate progeny E. coli cells. And so what they did was have an inducible system. So as you see in the right-hand side here, this IPTG induction of some of those archaeal lipid synthesis genes in a cell that does not have those archaeal lipids. You see the distribution of lipids in the cells are, we've got phosphatidylethylamine, phosphatidylglycerol here in green. This is the one with the G3 phosphate. And here we have cardiolipin. So this green one is going to be replaced by the archaeal version, the G1P backbone here in blue, as they add more IPTG to induce expression of those archaeal biosynthesis genes. And you can see with 10 micromolar, they were able to get nearly 30% of the archaeal lipid produced in place of that bacterial lipid. And so what they wanted to do right away was to see what does that even look like? What do those cells, what does the surface look like? What's the shape? And so over here on the right-hand side, you can see a cell that has just normal E. coli bacterial lipids. versus that which has had the archaeolipids induced. So you can see that the width remains around the same, but that the cells become very much elongated. So the cells are several times, maybe twice as long as the cells that have the bacterial lipids. And if you look a little more closely at the surface, it seems like it's a little wrinkled or pocked almost. What the researchers then wanted to investigate was whether the lipids from the archaea were able to add any type of temperature tolerance. So archaea can be thermo or extremophiles. They can live in extremely hot or extremely cold places more often than bacterial cells. And that doesn't mean that all archaea are extremophiles. We have archaea that are members of our microbiome, for example, and those are just archaeal cells that live in, our 37-degree body temperature conditions. But because they have, because some archaeal have these extremophile characteristics, the researchers wanted to look at the ability of these E. coli that had archaeal lipids to withstand high temperature and low temperature. And they saw that there was perhaps a small effect. So if we look at this panel A, we can see that the temperature is increasing along the x-axis, and here we have the number of cells that are able to be withstand this number of colony-forming units that are able to withstand this temperature and to grow at this temperature. And the temperature in these control strains here, the blue and the green, you can see that it diminishes quite quickly as we increase the temperature, with both cells able to have about 60 to 70% of cells recovering from temperature growth at 50 degrees. But that quickly after that, they poop out and no cells are able to grow once we hit 55 degrees Celsius. So if we go to look at the cells that have this archaeolipid, we see that they also have a similar growth pattern when we're at 50 degrees, but as we increase, they are able to have a few cells, a few colony-forming units that are able to withstand that 55 degrees temperature. But by the time we hit 58, those cells have also died. So there might be a very slight increase in temperature on the high end. The researchers also froze the cells at negative 80 degrees Celsius and then thawed them and looked at how many of those cells were able to withstand that freeze-thaw process. And they saw again that there was a very, very low percent of cells in all conditions that were able to withstand that, but it was maybe 4 to 5% of the control cells, and then those which had the induced archaeolipids had about twice that, maybe 12% or so of the cells were able to recover and grow again after that. So what this suggests is that maybe with a little more manipulation, bacterial cells could have archaeolipids that are engineered into their lipids and potentially give them the ability, these bacterial cells, to withstand different temperatures. And so this was a little bit surprising. The results were a little bit surprising to the researchers themselves. Science Daily quotes one of the co-authors as saying, "The robustness of these mixed cells surprised us. We expected more problems keeping them alive. After all, what we have engineered does amount to a new life form." And when you think about it, it is a new life form because the phospholipids have been changed in their orientation, in the way that those lipids are, the fatty acids are added, and the way that the glycerol has its modifications on it, which is going to change the way that the E. coli machinery interacts with that phospholipid bilayer. And so I thought this was a very interesting paper that gives us a little bit of insight into the potential for that last common ancestor to have had mixed lipid membranes. That doesn't answer the question, of course, but it does propose that there may have been the potential for a mixed lipid membrane. And then I included these two tweets because they were related a little bit tangentially to the paper. Marvel's Infinity War is coming up with all of the Marvel characters from the various storylines crossing over. There have been a lot of jokes on Twitter. And both of these researchers made the joke about Infinity War being the most ambitious crossover event in history, and then coming in with the crossover event of the archaea and the bacteria interacting to form eukarya, which is, as we talked about in another Microbial Minute session, I believe last year, one way that we believe that the eukarya were formed. All right, so we're going to move on to another PNAS paper, and this one is entitled Behaviors, Movements, and Transmission of Droplet-Mediated Respiratory Diseases During Transcontinental Airline Flights. There are a lot of different take-home messages from this research paper. The way that I've summarized it is that sick travelers are less likely to spread disease than sick crew. We'll talk about some other take-home points as well. So this is an important research study because the ability of travel via airplanes means that somebody who's sick with an infectious disease can hop onto an airplane and take that disease to virtually anywhere else in the world. And not only are they going to take that disease in their own body, but there's the possibility that they may sicken other people who are on the airplane with them, traveling with them to that new destination. And so the Fly Healthy Research Group wanted to look at the possibility of this happening. They did that by taking 10 different transcontinental flights in the US between Atlanta and various cities on the West Coast. They monitored the movement of people in the economy class. And I assume they did the economy class because those were the affordable tickets. And they also made sure to mention that these were I believe there are 737s and 747s, the type of airplanes that have 3 seats on both sides of one aisle going down the airplane. And so every time somebody moved in one of the seats, it was recorded by one of these researchers. And what they noted were that, as you might expect, people who sit in the aisle seats move the most. So as you see over here on the right-hand side, 57% of window seat passengers did not move during the course of that flight. And 48% of the middle seat passengers did not move. But only 20% of the aisle seat passengers stayed put for the entire duration of the flight. They would often get up to use the restroom or to get something out of the overhead compartment much more frequently than those who were in one of the seats that's blocked by that aisle. The researchers also wanted to look at the interaction of different passengers, and they saw that of the nearly 1,300 passengers that were monitored, 84% had close contact with another passenger seated more than 1 meter away. So that means something like speaking to another person, handing them something, somehow interacting with one of these other passengers. But of course, most of that interaction is going to happen between the passengers and the crew. So they were also monitoring where the crew was and how many times they interacted and how often they interacted with different passengers. The crew members were found to spend 1/3 of the flight time in contact with people, and that is another potential way for transmission to occur, either between the passenger and the crew member or the crew member and the passenger. So using all of these different data that they had accumulated, the researchers then modeled what would it look like if there were a sick passenger on the plane. If a passenger in seat 14A, which they just chose arbitrarily, were sick, how many other people would that person likely sicken? And so they looked at this as if it were a respiratory disease, something that would have to be spread by speaking or somehow coughing or some expelling of a virus. And they also did not consider fomites. So that would be something like a virus that was coughed out and landed on a seatbelt of the passenger. They only considered direct transmission between individuals. And so what they found is if they have a sick individual, as you have here, 14A, the triangle seat, those that are at highest risk of infection are, as you might expect, those that are closest to that particular passenger. So those on the other side of the aisle and all of those who are in the same section as that particular passenger, in that same row and in the row in front and behind as well. However, if you are even 2 rows away from that passenger, that risk decreases a lot. And so there's actually not a lot of risk. If you see somebody who's coughing on the airplane several rows away, there's no need to worry necessarily about transmission of that illness to you. The situation changed a little bit when it crew. Now, the researchers assumed that any crew member who was extremely sick would not come to work. Remember, this is their workplace. They don't want to spread disease to their coworkers or to the passengers. And so anyone who is extremely sick is not going to be at work. And so they assumed a slightly lower transmission rate between the crew and other individuals, because anyone who was extremely sick wouldn't be there. However, somebody who was extremely sick and had bought an airline ticket because of that expense that's paid would presumably take that flight anyways. So we see on the right-hand side that the risk of getting sick from a crew member is lower for everyone, but it also many more people are at risk. That risk is distributed but quite evenly between people who are in the aisle seats and the middle seats, right? With those who are in the window seats at the lowest risk of becoming sick from that crew member. And based on the transmission rates which they chose, the researchers predicted that one sick passenger is likely to infect at most one additional passenger. So that actually not a lot of transmission here between somebody who is flying who is sick. However, the sick crew member, because of the increased interaction between individuals, is likely to infect maybe 4 to 5 passengers through different interactions that would occur during that flight. Now, the research was done in part due to the documentation of over 40% transmission of influenza and SARS to people on planes. And so they were trying to reconcile what they had found with some of the previous data. And the researchers suggested that the airplane itself, the contained vehicle, is not the only place where transmission can occur when traveling. And they suggested that, for example, when boarding and handing a ticket to the gate attendant, for example, that is one source of transmission. Perhaps once you've arrived and are deplaning and picking up your luggage, that's another place where people are interacting, perhaps handing luggage from one person to another. And so it's not merely the act of flying, but the entire process of traveling, which can expose you to a number of other people and potentially lead to some of that transmission that was previously observed. So this was the exact type of article that is likely to be written up by mainstream news, and of course it was. There are a number of different take-home messages as written up in the headlines. So the LA Times writes, "To avoid germs on an airplane, consider booking a window seat." MarketWatch wrote, "Take this seat on an airplane to reduce your chances of getting sick." And the New York Times wrote, "How not to get sick on a plane, choose your seat wisely." And so all of these are emphasizing the ability to not get sick. Which kind of implies that there are seats that you can choose and become sick, right? And as you can see in the New York Times, they quoted one of the authors as saying, "It's pretty clear that if you're seated more than a meter away from an infected passenger, and if you're careful with hand hygiene, you're unlikely to get infected with the flu or these other respiratory diseases." And so that good hand hygiene means not only washing your hands frequently, but also avoiding touching one's face after handling other things. And you can see that the number of contacts was shared on social media, with the aisle, middle, and window seats showing the number of contacts they had with other passengers, with the most 0 contacts being found in the window seat. However, all of these, as I said, imply that there are seats that you can choose that might increase your chances of being sick. But I think one of the take-home messages, which is not getting a lot of play, is this from their finding, and that is that the team also collected samples from airplane surfaces before and after the passengers boarded. And they found that the airplanes were pretty clean. They swabbed the seat belts, thinking that the seat belts were the least likely area to be cleaned by the staff that cleans the planes at night. And they also looked at the trays and the armrests and other areas that might contain viruses that you could pick up. And of the 229 samples that were collected, not one had any evidence of the different respiratory viruses that they were screening for. So they were using molecular tests to look for 18 common respiratory pathogens, and they didn't find any evidence that these were present on the plane. So that means that the plane is a lot cleaner than perhaps you might think. I think that while it is true you can decrease your probability of getting sick by sitting in a window seat and by not moving from that window seat, it looks like a plane is not as likely to transmit disease as perhaps one would have guessed, at least certainly as I would have guessed, being trapped in with 100 other people for 3 or 4 hours. All right, so the last item that I want to discuss today is the budget that was just passed on Friday. The take-home message is that the congressional budget increases science budgets almost across the board. There's this lovely interactive graphic that you can find for yourself at the website listed below on AAAS. You can look at the different institutes. In this case, I'm highlighting the National Institutes of Health, where you can look at all the different areas of government funding to look at where the budget is for the 2018 budget. And I've highlighted a number of different science articles. So they published a number of really great articles summarizing some of the research increases. And so just to summarize what some of those numbers are, the NIH saw an increase of 8.3%. NASA science programs is 7.9%, with overall NASA increasing 5.6%. The USDAR's program had a 3% increase. US Geological Survey had a 6% increase and preserves all 8 agency climate centers, which is great. The NSF had a 4% increase, which may seem modest relative to the NIH, but will contribute quite a bit to a budget that's been stretched very thin. And in fact, in the bill, it's stated that this increase was in part because of the increase in basic research in China and other countries considered to be our competitors. The National Institute of Standards and Technology, increase of 4%. The DOE had a 16% increase, very, very large increase in their budget. And the only agency that I've found so far that had no increase is the EPA, which although did not have an increase, did not have a decrease either. And so it will remain budgeted as it had been previously. So across the board, this is just good news for science funding in general. We hope that a lot of this research will trickle down to the researchers who need it to fund their programs. And so I just want to give a short plug for the ASB&B podcast, Pipettes and Politics. They haven't, as far as I know, spoken about this particular budget yet, but they do a great job about talking about science policy and the impacts that it has on different research aspects. And so we'll include a link to that below as well. And ASM also put out a note congratulating Congress on increasing this important funding. ASM President Peggy Cotter was quoted as saying, "Today, Congress made a strong federal investment in programs and initiatives that secure our borders against natural and intentional biothreats, ensure a healthy public, and retain our global leadership in advancing science." So a nice, happy note to the congressional budget. I'd like to encourage you to subscribe if you're interested in this type of update every couple of weeks. I'm going to be back in another couple of weeks with what's hot in the microbial sciences. I'll check the chat box to see if there's been any questions at this point. But I'm afraid that we are out of time and I have another meeting to get to. So thank you very much. If you do have a question and I didn't get to it, please leave it in the comments below. And I will talk to you next time. Just to summarize what we did talk about this week, we found that 1/4 of non-antibiotics affect gut microbiome members, that artificial intelligence IDs new viruses, E. coli lipid experiments give insight into the LUCA membrane. Sick travelers are less likely to spread disease than the sick crew, and that the congressional budget increases the science budgets of many of the national institutes. Thank you very much, and I'll speak with you next time on microbial.